Hi, welcome to the second session on FMD Diagnostics. My name is Michael Eschbaumer of the National FMD Reference Lab at the Friedrich Loeffler Institute in Germany. And it's my pleasure to chair this session together with Phaedra Blay of the Dutch NRL at Wageningen Bioveterinary Research in Lelystad. We have two very different talks today, but I think that nicely demonstrates the diversity of diagnostic activities that are necessary for effective disease control and prevention. The first talk is going to be by Petras Janssen van Vuuren of the Australian Centre for Disease Preparedness in Geelong, which you may still know by its old name, the Australian Animal Health Laboratory. Their research that is presented today addresses an important issue for foot and mouth disease diagnostic activities in the context of an outbreak in a free region or anywhere really. In these situations, not only the quality of diagnostic results and the speed with which they can be obtained is important, but it's also essential not to contribute to the spread of the disease with your diagnostic activities. This includes the people taking the samples, their vehicles and equipment, but also the samples themselves. While vesicular epithelium is the most useful sample matrix due to its high viral content, this also makes it particularly dangerous to handle, ship, and test in the laboratory. So when you collect samples on a farm that is under an FMD suspicion, what can you do to reduce the risk that is associated with the transport of these samples to the lab and with the diagnostic tests that will be carried out there? The most important feature of protocols and procedures that are useful in this situation may be the efficacy and the kinetics of the virus inactivation itself, but it doesn't end there. Is the procedure easy to do? Is it safe for the operator to do? How complicated are the logistics of the required reagents, etc.? So while some chemicals are easy and cheap to procure in advance and can be stored indefinitely while you're waiting to have them at hand when the outbreak occurs, others may be more costly, may only be available from one supplier or not available in all markets, not shelf stable, and many other issues that can arise. At the same time, reliable and quick inactivation alone is not useful unless you can be sure that whatever is being done to make these samples safe for shipping and handling does not interfere with the sensitivity and specificity of the diagnostic procedures that you want to do once the samples do reach the laboratory. So I think that these are some of the questions that will be addressed in our first talk in this session. And I'm particularly happy to have Petras here today because the research that he's going to present comes from an ongoing collaboration between the group of Vilna-Voslo in Geelong and my laboratory at the FLI. GFRA is an excellent forum to create and promote this kind of international collaboration. And I think this research is a great example of that. So that's it from me. Phaedra, can you introduce our second speaker, please?
Yes, sure. I'm happy to co-host this session with you, and also happy to do it on behalf of the GFRA. And thank you for the introduction of the first topic in this session. And as Michael already indicated, the area of diagnostics is very wide. And so the second presentation in this mini session will deal with a totally different topic in this area and concerns the development of a new ELISA. And despite all molecular techniques that are being used nowadays, ELISAs are still very important tests that are being used in our labs, both for antigen and antibody detection. And serological tests for detection of FMDV antibodies are needed for either epidemiological surveys, assessment of vaccine efficacy, or in order to prove the absence of these antibodies. And at the first day of our meeting, Alejandra has given a very nice overview of several serological assessments of FMDV antibodies. And one very well-established serological test is, of course, the liquid phase blocking ELISA for the detection of FMDV antibodies, which was developed a long time ago by Chris Hamlin in 1986. This test has undergone changes in time, but still in this ELISA, polyclonal antibodies of rabbits and guinea pigs are usually being used. And in diagnostic tests, apart from the use of polyclonal antibodies, monoclonal antibodies can and are being used. But the relatively new type of antibodies that also exist are single domain antibodies, or VHHs, which are produced by camelids. These VHHs are very different as compared to other antibodies because they do not have a light chain. Furthermore, these VHHs have a high stability and solubility and can be well expressed and produced in microorganisms. And these specific features makes them very suitable to be used for many different purposes. And for example, VHHs are being used for therapeutic applications in human cancer therapy. VHHs have also been used in foot and mouth disease research, and as far as I know, they have been used for passive immunization studies and in ELISAs that are used for vaccine quality control. In these tests, VHHs that can specifically detect 146S or 12X particles play a role. And I think Joaquin will present his work on an ELISA in which the VHHs are used in a liquid phase blocking ELISA. So this second presentation will be presented by Joaquin Bosso of the Centro de Virología Animal in Argentina. And he told me just a few minutes ago that it's part of a thesis that he is going to finalize, which has the VHHs as a main topic. And I'm really interested in also the other work of his thesis. So that's it. Over to you, Michael.
Thank you, Phaedra. Thanks. As usual, please put your questions for the speakers in the chat. And now, Pedros, the floor is yours.
Good day. I would like to thank the organizers of the GFRA 2021 conference for allowing me to present our work on safe transport of epithelium samples infected with foot and mouth disease virus. Diagnostic laboratories are classified according to their biocontainment level. The World Health Organization designates laboratories as BSL 1, 2, 3, or 4, where the basic laboratories at BSL 1 and 2 perform basic teaching and research or primary healthcare services, diagnostics and research. Containment or BSL 3 laboratories perform specialized diagnostics and research on pathogens that present a major risk to human and animal health and for which effective treatment or preventative measures are available. Maximum containment laboratories or BSL 4 laboratories handle the most dangerous pathogens for which there is no effective treatment or preventative measures readily available. In FMD-free countries, diagnostics for FMD is performed at BSL 3. However, most countries only have a limited number of these laboratories available because of the high cost of constructing and maintaining them. During outbreaks, heightened surveillance may, however, overburden these limited number of BSL 3 labs, and therefore low-risk samples may need to be sent to BSL 2 labs. FMD virus is acid labile and easily inactivated at pH lower than 6.8 in low ionic strength buffers. However, when this virus is protected in biological matter, the virus can be resistant to inactivation. For example, it has been shown in slurry that the virus can remain infectious up to 9 days at 20 degrees Celsius and 14 weeks at 5 degrees Celsius. In other matrices like tissue, the virus can survive up to 10 or 77 days, depending on the temperature. In a previous study, commercially available lysis buffers were tested by incubation at room temperature for 10 minutes with virus. These buffers were shown to completely inactivate the virus. However, in this case, spiked epithelium tissue suspension was used instead of tissue from experimentally infected animals. There are a number of factors that impact the effectiveness of virus inactivation. These include the virus titer and strain, contact time, reaction temperature, the concentration and composition of the lysis buffer, and very importantly, the sample matrix. Most experimental studies so far have only used one virus strain. but care should be taken to extrapolate to all FMDV strains. Also, spiking tissue samples with virus also does not fully represent how the virus will react in infected tissues from animals. Therefore, we set out to investigate whether FMDV in epithelium tissue will be inactivated using a number of commercially available buffers that also offer the additional advantage of preserving RNA. Following that, we wanted to determine if the virus can be recovered from the genetic material isolated from these tissues that were inactivated in these buffers to allow further investigation under suitable biocontainment levels. We inoculated 2 groups of 4 cattle each via the intradermal lingual route using an Indian 2001 lineage virus from serotype O and A Asia genotype 7 virus. Tongue and hoof lesions were collected on day 2 after inoculation, and we evaluated 5 inactivation buffers and compared that to 1 control buffer. We performed manual homogenization and compared that to passive incubation of the tissue in the buffer. We wanted to evaluate virus inactivation over time and how well the RNA was preserved for further downstream processing. We collected tissues of 20 to 25 square millimeters into buffers including PBS, RNA later, DNA and RNA shield, the 2 separate components of the PAX gene system, namely the fixative and the stabilizer, as well as a very simple buffer, McIlvaine citrate phosphate buffer at pH 2.6. For the manual homogenization, we used a plastic pestle in an Eppendorf tube. We then incubated these at room temperature for 2, 6, 24, and 48 hours. We then performed virus isolation on these homogenates using 24 well plates and LFBK cells. We performed 2 passages, with the second passage only performed when the first passage did not yield noticeable CPE. The DNA RNA shield was found to be cytotoxic, and therefore we had to perform an amended isolation protocol using larger flasks and diluted sample to prevent the effect of cytotoxicity. We performed TCR50 titration also using LFBK cells and real-time qRT-PCR that targets the 3D pol coding region. Sequencing was performed using primers that target the VP1 coding region. And transfection was performed using lipofectamine 3000 and LFBK cells. We showed that RNA later yielded variable inactivation results depending on the virus type. DNA and RNA shield seemingly inactivated the virus, but there was also some level of toxicity. And upon further investigation using larger flasks, we showed that there was actually replicating virus in samples up to 6 hours post-incubation. The RNA yield, as measured by qRT-PCR, was comparable between all the buffer types. Similar to the results with RNA later, the PAX gene stabilizer component yielded variable results depending on the type of virus used, with O virus being inactivated relatively quickly, but requiring 24 hours to 48 hours for inactivation with the A-type virus. The fixative component was more effective at inactivating virus, but still required more than 2 hours for complete inactivation. The citrate phosphate buffer, McIlvaine buffer, was very effective at inactivating both viruses within 2 hours. As with the other buffers, the yield of RNA, as measured by qRT-PCR, was comparable between the buffers. We found that manual homogenization prior to incubation in buffers had no effect on virus recovery or RNA preservation. We also showed that virus recovery was successful by transfection with RNA from epithelium that was incubated in all the preservation buffers. Likewise, VP1 sequencing was successful with RNA from epithelium in all the preservation buffers. In conclusion, we showed that RNA later in PAX gene RNA stabilizer component is likely unsuitable for rapid inactivation of foot-and-mouth disease virus in epithelium tissue, since it required more than 6 hours incubation for complete inactivation, and there was also variability depending on the type of virus used. DNA and RNA shield was shown to require between 2 and 6 hours incubation for inactivation. This is a much better result, however, might still not be suitable for on-farm inactivation of virus in epithelium tissues before transport. The fixative component of PAX gene was shown to require at least 2 hours of inactivation. However, the PAX gene system is quite expensive and might not be suitable for this purpose either. McIlvaine citrate phosphate buffer at pH 2.6 is cheap and easy to produce and was shown to completely inactivate the FMDV virus in epithelium tissue in less than 2 hours. We further showed that all the buffers in the study was able to preserve RNA following at least 48 hours incubation. However, we don't know the effect on RNA integrity with longer incubation. It should be feasible, however, for samples to reach laboratories from on-farm collection within 48 hours in FMDV-free countries. We showed that McIlvaine buffer is the simplest, cheapest, and most effective option for on-farm inactivation to maintain biosecurity at key testing laboratories in FMDV-free countries. Inactivation of FMDV in epithelium tissue is therefore possible using very simple and cheap to produce buffers, and that would allow safe transport between the farm and laboratories, and thereby maintaining the biosecurity of biosafety level 3 and 2 laboratories in FMDV-free countries. I would like to thank you and acknowledge the funders and the co-authors on this presentation. Thank you.
Hello, my name is Joaquín Bozo, and today I'm going to talk about my work, feasibility studies of camelid-derived single-domain antibodies as capture antibodies in a liquid phase blocking ELISA to detect antibodies elicited after foot-and-mouth disease vaccination. This work has been done in the Centro de Virología Animal and in the Instituto de Ciencia y Tecnología Dr. César Milstein in Argentina. In addition to conventional antibodies, camelids also present the so-called heavy chain antibodies. They lack the light chain and the CH1 domain. So the antigen binding site consists of a single domain called VHH. It is possible to obtain this VHH by recombinant DNA technology and express it soluble, keeping its antigen binding properties. VHH on antibodies have many advantages when compared to conventional antibodies or even to other recombinant ones. They are easy to produce in prokaryotic or yeast expression systems. They reach high yields of expression. They are highly stable and soluble, and they are easy to directionally immobilize to solid supports by C-terminal peptide extension. Regarding foot-and-mouth disease, in Argentina, annual vaccination campaigns are performed, and since the year 2005, Argentina has applied a tetravalent inactivated virus vaccine with antigen corresponding to strains O1 Campos, A24 Cruzeiro, A Argentina 2001, and C3 Indayal. This vaccine must be controlled batch to batch, and among these controls there is the indirect potency test, which is a serological test performed to measure the titers of elicited antibodies after vaccination. Liquid phase blocking ELISA is an assay performed to control the vaccine potency, where the measured antibody titers correlate with protection conferred by the vaccine. This kind of ELISA consists of a liquid phase where the antigen corresponding to one of the strains is incubated with serial dilutions of serum samples from vaccinated animals, and a solid phase where a homologous rabbit serum is coated on a plate well as capture antibodies. These two phases are incubated together, allowing the capture of the antigen if it is not previously blocked by specific serum antibodies. If the antigen is captured, a specific monoclonal antibody would bind to it, and then an HRP conjugated antibody. As a result, low antibody titers will develop high signal, while high antibody titers will develop a low signal. After assaying 4 serial dilutions, the titer is expressed as the reciprocal logarithm of serum dilution, giving 50% of the signal recorded in no serum control, which would be this case, the 100%. Given the properties and advantages that make nanobodies suitable as detection probes, we hypothesize that in comparison to polyclonal serum, the usage of VHH as capture antibodies in ELISA could increase batch-to-batch reproducibility and simplify scaling up the production of identical antibodies. So we aim to study the usage of specific O1 campus VHH as capture antibodies in a liquid phase ELISA in order to measure antibody titers elicited by anti-foot-and-mouth disease vaccination. VHHs were previously obtained. First, we performed the LAM immunization with the tetravalent vaccine containing the X4 strains. And after confirming a high humoral response, the VHH gene fragments from lymphocytes were amplified by PCR, ligated in a phage display vector, and transformed into bacteria to construct an immune VHH library. Then VHHs were selected by solid phase phage display methodology against O1 campus strain antigen, and finally, we screened a total of 182 selected VHHs, and their activity was confirmed in both ELISA and Western blot. As a result, 28 VHHs turned out to be O1 campus specific, and the 4 most reactive VHHs were further studied as capture antibodies. First, we performed a capture VHH ELISA to see if these 4 VHHs, 2F1, 5A1, 5B4, and 11G2, retained their specific O1 campus reactivity when immobilized on a solid phase. The immobilized VHHs were incubated with these 4 strains separately, and detection was performed with a monoclonal antibody. We compared this capture with a non-related VHH, the homologous O1 campus rabbit serum, and the homologous sera for its strain. And in this figure, we can see that all 4 VHHs only capture O1 campus strain antigen, showing a more specific capture than the O1 campus homologous rabbit serum as expected. Once we confirmed that the antigen could be captured by these 4 VHHs, we aimed to study the optimal VHH mass for coating. A capture VHH ELISA was done, immobilizing different VHH amounts per well. In this figure, we can see in the y-axis the absorbance, and in the x-axis the VHH mass per well, and how the absorbance rises and then remains constant at 250 nanograms per well, which is expected by previous reports. When comparing the antibodies, VHHs 2F1, 5A1, and 11G2 were the ones with the highest capture levels, so we kept studying them for their application in the liquid phase blocking ELISA. After determining the optimal mass for well coating, we compared the antigen capture using different antigen dilutions. In this figure, the absorbance is shown in the y-axis and the inverse of antigen dilution in the x-axis. We studied the capture of VHHs 2F1, 5A1, 11G2, and the pool of these 3 VHHs, and the homologous rabbit serum for O1 campus. Among these VHHs, the highest antigen capture was observed for VHH 2F1, so this VHH was selected as the nanobody to be used in a VHH liquid phase ELISA as a capture antibody. To compare the performance between the conventional rabbit serum liquid phase ELISA and the VHH liquid phase ELISA, we assayed 81 sample sera from vaccinated bovines. As I explained before, dilution of these sera samples were incubated with O1 campus viral antigen in a liquid phase, and the mix was incubated with a solid phase. In one hand, the immobilized rabbit serum, and the other hand, VHH 2F1. Then, we compared the titers measured by each techniques. The comparison consisted in determining the concordance between both ELISAs. As shown in this first figure, a direct comparison was performed. On the x-axis are represented the results of the rabbit serum liquid phase ELISA, and on the y-axis the VHH liquid phase ELISA. The blue line depicts the line of perfect agreement, and each dot represents a serum titer determination. As we can see, the point cloud is superposed to the blue line as expected when there is a good concordance. The concordance correlation coefficient, or CCC, was calculated as a measure of concordance. The resulting value was 0.97, very near to 1, thus indicating a very high concordance. To further study this high correlation, a Bland-Altman diagram was plotted. In this figure, the x-axis shows the average of measured titers by both techniques for each sample. The y-axis shows the difference between the titers. The red dotted line indicates the mean of all difference, which is nearly zero, thus indicates that there is no systematic deviation between measured values. The gray lines indicate the limit of agreement, which is 0.2. This means that in 95% of the determinations, the difference between both techniques would be less than 0.2. So we conclude that there is a high concordance between the VHH liquid phase ELISA and the conventional liquid phase ELISA. As a conclusion, and to summarize all I have presented, the study of VHH only captured O1 campus strain when used as capture antibody in ELISA. The optimal mass for VHH well coating was determined as 250 nanograms per well. The VHH2F1 presented the highest capture performance, and a high concordance was observed between both liquid phase ELISAs. Finally, these results indicates that VHH liquid phase ELISA has the potential to be used to detect antibodies elicited in bovines by foot and mouth disease virus vaccines.
Thank you very much to our presenters. I do not see any questions in the chat yet, so maybe I can just start with a question of my own. So, Petros, you already saw some serotype specific differences in with some of the buffers that you used, even though there were only 2 viruses in this study. So what does that mean for being able to apply your results to like all of FMDV in the world?
I think it's too early to apply this to any virus. I think we might need to do a more expansive study using more viruses, not only viruses from all the serotypes of concern, but also maybe some key viruses from key lineages and sublineages within those serotypes. So I think this is a good start, but we do need to confirm that whatever buffer we identify from those as the best, that it does actually work against a good amount of viruses that are currently circulating.
Mm-hmm. Okay, thank you. Phaedra, do you have any questions?
Yes, I do have actually several questions for Joaquin. Thank you, both of you, for your presentations. And I was wondering, you immunized llamas with several serotypes in the vaccines, and do you intend to start producing ELISAs for the other serotypes as well? And related question is, do you aim to use VHH as a second antibody too in these ELISA? Do you think that would be feasible?
Well, addressing the first question, when we did the solid phase selection by fake display, we end up with 2 groups of VHHs, the O1 campus specific VHHs, and we also had a cross-reactive group, which showed reactivity for the other 3 strains. So now we are working with these cross-reactive VHHs to perform a liquid phase ELISA using these cross-reactive VHHs. So we can use this kind of VHHs to capture the 4 antigen strains. And the second question was regarding the usage of the VHHs as detector antibodies. Yes, we would like to say that, but it's not something we have started.
Okay, I see now questions coming in the chat box, also for Joaquin. It's from Maria Cruz Miraglia. Could you already test the other VHHs produced? It says A24, A2001, and C3. I think you commented already on that. And do you know which epitope is recognized by your VHHs?
Regarding the last question, no, we couldn't know which is the epitope recognized by this specific VHH, the 12-1. We try to characterize their activity, for example, in Western blot, to at least see which of the BPs was the protein recognized, but not all VHHs are reactive to a linear epitope, so we couldn't see reactivity in a Western blot. So the answer for the last question would be no. And the first question, I don't know if I understand it. The question is if I have already tested these VHHs as liquid phase, as capturing a liquid phase.
It says, have you already tested other VHHs produced? And it refers to the serotypes A24, A2001, and C3.
Yes, if I could, I could. I don't know. Sorry, maybe I'm not understanding the question.
That's not a problem. There's many other questions that you can take a stab at. I just looking at the chat. Other people have asked whether you know whether your antibodies specifically recognize 146S particles, or do they also recognize 12S?
They all recognized 146S and 12S. There are works where they perform a specific selection for 140S, but in this case, the VHH we used, the VHH2F1, recognizes 140S and 12S.
And did you try your VHH antibodies in Western blot?
Yes, yes. As I said before, yes, we assayed it. They are not all reactive in Western blot. That's maybe because of the kind of epitope that it recognizes. If it's a conformational epitope, it probably won't be reactive in a Western blot. But there are some others that they can recognize VPs on Western blot.
There's a follow-up question. Aldo asks, did you try the Western blot in non-reducing conditions? He thinks you might find more reactions if you did that.
Yes, no, we didn't try that, but yes, it would be a good approach to see which protein its DBHH is recognizing.
There's, I think, one question that we missed so far. It's a little bit further up. Alejandra asked, did you already try conjugating the VHH? Because that may be useful because you're using two different antibodies, the detector and the conjugate?
Yes, I think the question refers to the use of VHH as detector proofs. And in that case, we have everything to do that, but we haven't started the conjugation of VHH to HRP. That would be the strategy.
Okay, thank you. We're being informed that our allotted time is up, and I think if anybody else has other questions for our speakers, they could continue the discussion in Gathertown during the coffee break. Phaedra, do you have anything to add?
No, thank you. I would like to thank again both presenters and all the other persons for listening, and hope to see you in Gathertown.
Thank you very much.
Thank you.